Good morning. My name is Jiyoung from the Department, from the Department of Food Science and Technology. I also have a split appointment, College of Public Health at the Division of Environmental Health Sciences. So I'm an environmental microbiologist. Today's talk about harmful algal bloom, HABs, but switching gear to more impact on our health, but also because this is CFAES conference, I'm mostly focusing on food safety and food security. Okay, as a microbiologist, my research focus has been microbes and their metabolites, mostly toxins, and what happened to us when we exposed to, what are the health outcomes. So that is an example of what we did, the beach water, the bacterial contamination and gastrointestinal disease, and also drinking water, and then how the bacteria from drinking water colonize in our gut using germ-free mice. And so pathogens to all the way to health outcome, there are many ways to do, but first one, we need to understand pathways of exposure and transmission dynamics. So my major focus on enteric pathogens and cyanobacteria and toxins, but also we are looking at metagenomes and registome. So all the water we are looking at, actually there's one water, drinking water all the way to wastewater, but today's focus only on cyanobacteria and toxins. So we saw this picture many times today. So that's the Lake Erie, but Lake Erie is not the only one. That's a universal global problem, including South Korea, China, South America. But the problem is we have more in higher intensity, more frequency. The problem is the cyanobacteria that make toxin. So we are talking about microcystin because that's the most prevalent toxin, which is a liver toxin. So I've built gradually a lot of collaboration across the campus to see this kind of research question. So all the way to cellular remote sensing, epidemiologist, but also Jay Martin from FABI Ecological Engineering and fish person, and then drone to monitor more closely the water, but also what are the chronic health effects, looking at carcinogenesis. Okay, so this is the main talk, so public health and cyanobacteria bloom across the US. So we did across the US human health data, especially non-alcohol liver disease, and the bloom extent using remote sensing. So we analyzed more than 10-year data linking both human health and then blooms. So the map shows that 63% of our county had detectable cyanobacteria bloom coverage. So that's the bloom and the liver damage. So liver disease, the red color means high bloom, high liver disease. Blue means low bloom and then low liver disease. So those blue and red are the significant for further analysis. So what we found was there is a statistically significant association between bloom coverage and the liver disease. And then that means if we have 1% increase of a bloom coverage, that leads to 0.3% more liver disease. So then we need to talk about the exposure. The water is the one, but how about the food? So fish is the one, but how about the fresh produce? So that's the study we've been conducting for the past couple of years. And then the recent finding was that the irrigation water, if we have toxins, it can go to our crops and then also the soil. So that's the recent publication. And then that was published last year. But that toxin is accumulated, but also it can accumulate in the soil, but not just the human health impact, but also the soil can be a problem. So where does the toxin go? So the green means it goes to the plant, and then brown means it goes to the soil. So if we add more and more toxin, actually there's dose-response relationship. So what does that mean in terms of potential health risk? So we estimated intake and then how much we consume per day in terms of lettuce, and then we also tested carrots. Okay, so the children, adult, we estimated And then 1 ppb is the drinking water, and now it's a little lower than drinking water guideline, and then 5 ppb and 10 ppb, and then 1 ppb is still moderate health risk. But how about the crops? So, how about, okay. So crop, that's the example. So you can see the comparison between control, and then when we add more and more toxin, actually the quality is really deteriorates in terms of mass and then diameter. So it's not just food safety, but also food quality, and then food security can be an issue. So we build more and more interdisciplinary team to see this HAB-related research. So a lot of things are very dependent on how we accurately measure. So I collaboration with our LCMS team. And then also, how about the tide drainage? How about the smaller lakes rather than Lake Erie? So I collaborate with Kenyon College team, and then also how we can solve the problem. So one is we can use wetland, so that is the kind of one of the solution. So how about the future goal? So I think with all the expertise across the campus, my vision will be the one kind of center, so we can do HAB research together and in a more innovation way, but also foster the collaboration, and then also kind of talking with our state agencies. So that's all I have. I can talk more during our kind of break. Thank you.